are more on that side, just let me know. We can eventually discuss after the talk in details. So the talk is about, developing interfaces, systems, you can think, for remote monitoring of metabolism. I'm sure you know, sorry. Voila. I'm sure you all think why that, we try to transform our personal electronics in what someone called a distributed diagnostic tools by adding electrochemical sensor, dedicated electronic front ends in order to exploit our personal electronics already with us as a diagnostic tools. One of the latest news I love is this button from Hubbard that I'm sure you have seen on the beaches, on the airport during summer. So it's a little, it looks like wearable. Actually, it costs microneedle that break the skin in order to collect under the skin. It also, of course, electrochemical sensor and electronics, and then there is, a user interface. So the market is thought to going to explode. This is a quite old forecast that thought about 17 billion by 2025. Actually, more recent upgrade confirmed 61 USD billions worldwide in 2022, and the idea is to move from 3 billion devices to 26 billion devices connected by 2030, so an enormous amount of devices. And among them, you see 3/4 are for our customer electronics and health monitoring. 1/3 is about just health monitoring. So it's an enormous work to be developed from now to 2030. So the vision we're targeting is this one. So to have a fully connected humans. The point is that if you look closer, some of this stuff are already in the market, ECG or ECG or blood pressure. But if we look at the molecular level, toxin, DNA, proteins, we are not really already there. So nothing in the market just by glucose monitoring, while we have plenty of molecules we would like to measure to address many others. So along this track, what we usually need is the full integration of biomaterial in order to be selective if I want to measure one molecule out of millions of molecules in the bloodstream. I will show you cases. Sometimes we have the sensor, but it's not sensitive enough. So sometimes we need help by nanomaterial in order to improve the performance to reach the application. And if we want to have autonomy in the system, if we want just to carry a wearable and forget about that, we need autonomy. That's the reason we need electronics. I usually like to highlight in this slide 3 keywords, Bio, Nano, and CMOS, because this drove me many years ago now to the new concept of a co-design. Not to design the electronics and look for the application, not to design the sensor and then look for electronic to be coupled, but to co-design the whole chain of the system in order to succeed. I have even published a book, first edition more than 10 years ago, last edition out this year. So if you're interested, have a look of the content. The plan today is to show you some hits about the Nano, the Bio, and as I said, I reduced it as much possible, the electronics, and then I will go to applications that probably is more interesting for you. So what's about Bio? So we can measure, as I mentioned, many molecules. We have the needs to measure many molecules, including drugs. If I want to address precision medicine, I need to identify the right dose and the right drug for that particular patient. Believe me, I had a couple of projects in anesthesiology. There is a 200 standard deviation patients by patient. So the average numbers say nothing about that patient. So in principle, we would like to measure the molecule in circulating in that moment on its blood and then to adjust the dose. So how to do it? There are many different molecules we can detect that correspond to many different detection methods. I will just show you one case that is probably less trivial than others, how to measure drugs with electrochemical sensor. So there are, for the glucose, the lactate, the glutamate, there are the equivalent oxidases. I'm sure many of you knows about that. For the drugs, we can exploit another protein family, the one of the cytochrome P450. More than 3,000 isoforms, so plenty of choices. To understand how they work, just let us consider one example. If I have the bensphetamine circulating on me, is a drug for the mind. in my body, the cytochrome P450 2B4, one out of these more than 1,000 isoform, will catalyze the bensphetamine by consuming oxygen and receiving help from the coenzyme NADPH and consuming protons. This is typically done by my body, by body of mammalians, because the oxidized form is more soluble and faster to be secreted. Never forget that any exogenous pill is felt by our body as an alien, and they immediately start to push it out. The phenomenon in pharmacology is called clearance, and that's the reason we need to retake the pill after some hours. But for our aim, if we look closer here, it's a redox reaction. The coenzymes just furnish electrons. So in principle, we can furnish electrons with our electronics systems and then business, because we measure a current that corresponds to how many concentration of the drug we had in the patient's blood. Another interesting case is the one of ions. Years ago, we collaborated even with Mehmet and Ali on this field. In this case, we can use measurements of open circuit potential. If, for example, in this cartoon, potassium goes up or goes down, we have variation in the potential we measure at the interface. However, as the cartoon show, we have typically very much drift, and this is a very well-known phenomena all around the world. I will show you in a while how we could solve the problem of time drifting in ion sensing by open circuit potential. Exactly. What's about nano? How the nano can help us in drug detection or in ion sensing? Well, this is one example from the literature, the detection of verapamil, a very well-known antihypertensive drug, detected by another isoform through the 3A4 of the cytochrome P450. You see the calibration curve is quite good. We have a linear relationship between concentrations and currents measured. However, we need to see how much is the concentration in patient's blood. Below 0.3 micromolar. No way to have a real technology based on that sensor, because we are several orders of magnitudes blind with respect to what we need to measure in the patient's blood. But even though this is repeated for many other compounds, this is the case of the cyclophosphamide, a very well-known anti-cancer compound detected by exactly the same isoform of the cytochrome P450. You see, if I try to just use the enzyme, I have a linear calibration, but the calibration exactly starts when the therapeutic range ends. So again, I'm too blind for a real application. But if I introduce multi-wallet carbon nanotube in my interface, I fairly well succeed in targeting the concentration in the patients. We have tried and repeated with Phthoraful, etoposide, and other anti-cancer drugs, and it works always. So you see, in this case, carbon nanotube brought us back in business with respect to a real application. How to integrate in our electronic system? Well, one way that we can do is to directly grow carbon nanotube on our platform. So let's imagine to have a multi-panel device with many sensors, many working electrodes. When I want to have my carbon nanotube, what I can do is to flux, to pre-deposit a catalyst by electrochemistry, and then flux carbon-based gases. I can grow nanomaterial on top of my multi-panel devices. Another bad news is that I need to do it at 700 degrees. If I do it on CMOS, I'm burning all my electronics. So no way again to do it like that. But years ago, I had a quite clever students that also come for a period to the Ali labs in Boston, Arina Taurino, and she developed a smarter way to try to set down the temperature and goes up again with a grow by successive CVD deposition. So we can do it, but it's not that easy. If we follow the Irene process, we can even decide which kind of nanostructure grow on top of the multi-panel just by starting by different quality of the precursors deposition. We can have from fully compact to very rare nanoparticle of the precursor, in this example, the iron. And then depending on which is the quality of the precursor, we can grow carbon nanotube or nano petals, kind of rose flowers, where every petal is done by graphene layer. And you see here in the example of the glucose detection, we goes up with the nanomaterial on the electrode, and we goes up with the sensitivity of our sensor. So we are, again, back in business by a direct deposition, direct growth or carbon nanotube instead of just spreading, as in the example of cyclophosphamide I showed you before. As I mentioned, this is possible to be done on electronics, but it's tough. We run the risk to burn because we are approaching 450 degrees. So what's about alternative? For example, nano platinum or nano gold. It's very easy to grow directly on working electrode just by electrode deposition from salts. If we use the valent salts of platinum, we can get nanosphere. If we use tetravalent, we can easily obtain nanowalls. And we can even do combined deposition. It's again Irene work done when she was doing PhD in my lab. You see nanoparticles, and then on top of nanoparticles created kind of nanowalls. And again, we can use this material for glucose detection, for whatever other metabolite detection. And we can now easily integrate it on CMOS die. This is a collaboration I've done years ago with Imperial College, my good friend Pandelis Georgiou. Sarah Goresi-Zadeh was that time a PhD of mine visiting in London, and you see we designed the electrochemical function in the CMOS, and then after receiving the chip from the foundry, we have created by electrochemical deposition balls and nano walls directly on top of the exposed metal in the CMOS die. With this method, we have also solved the time drift of the ion sensing, because it is known in literature that the potential is drifting because there is a water layer in between the selective membranes and the electrode. And it seems that if we are nanostructured in the electrode, this water layer gone, and you can see here clearly how the time stability is highly improved by using nanopetal and nanosphere. So thanks to nanostructure in the electrode, also the ion sensing became much more reliable. What's about the electronics? So there are many different methods that we have developed over the year. As I promised you, I skipped all these details because you're probably less interested than the chemical details of the application. So just a little, overview on several chips that we have developed over the year. And then I will show you some example of how we have used this chip for electrochemical sensing. For example, in translational medicine, we have integrated everything in an implantable device. You see the last coil. I will show you in a while the detail why there is a coil there in the peritoneal region of a mouse, and then the mouse was free moving in the cage, and we were constantly monitoring its performance. With this chip, we have measured the glucose and the lactate. You see here calibration compared with Autolab, which is a kind of Ferrari instruments in any electrochemical lab. You see fairly good comparison between our C and the Autolab. Of course, we pay the price of worst limit of detection because our chip was, 1.5 millimeter by 1.5 millimeter, not that much filter inside. The Autolab is a machinery bigger like this desk with a lot of filtering. So clearly setting down the noise means setting up. Setting down again, that means setting up as a quality the limit of detection. By the way, lactate and glucose are in the millimolar range of concentration in biological systems. So a limit of detection of 80 or 100 micromolar is more than sufficient for the application. In case of drug detection, I'm sorry I have no time to show you details, but we really need to measure the Faraday peak. It's not sufficient to play in chronon pyrometry usually. So the comparison was done with the etoposide, very well-known anti-cancer compounds. You see two peaks very well detected, both with the Autolab and with our electronics. So it seems we are in business. Again, here the issue was to find correctly where is the position of the peak, changing the concentration, for example. You see the comparison is fairly good between reference lab instrumentation and our electronics. So what's about application? What we have done with all this magic over the last years in EPFL? So we have, of course, applied first to implantable and to wearable. So we have developed both portable devices, wearable devices, and implantable devices. And I will show you some examples. So for example, in case of portable, this is a device for the intensive care units developed years ago in collaboration with the CHUV hospital, main hospital we have in Lausanne, one of the main hospitals in Switzerland. You see probably some yellow pieces. Every yellow piece is an electrode. I'm sorry, but the pointer doesn't work on your screen. And then underneath there is the electronics for the communication. And you see on the cap, on the top cap, maybe I can, okay. You see here, these are the electrodes. This is, of course, is the integrated electronics. And there is a packaging that integrated fluidics. So the idea was to cut a draining tubing from the patients and put this little box in between to monitor typically parameter of intensive care units at molecular level. So for example, glucose and lactate. Glucose, you know, is part of the feeding of the cell, so it's an important parameter to monitor. Lactate is a marker of suffering of cells. So lactate is going up when typically the cells are suffering. So in case of intensive care unit patients, clearly the patient is suffering, clearly his organs are suffering, the cells are suffering, so lactate became an important parameter. This was another experiment done on the same application, is potassium measurements. Potassium is another marker of the fact that of organ failure. So if the organ fail, the cell died and they release a lot of potassium. So you see here a huge wave of potassium. In this case, it's a simulated organ failure. So we have put the cells to apoptosis. So we have let the system to measure the wave of potassium released by the cell during going apoptosis. Other applications were in anesthesiology. I already mentioned you the large variation patients by patient. So years ago, we were required by the CHUV hospital in Lausanne to develop technology, start in developing technology to monitor anesthesiology. A typical anesthesiology cocktail foresees in many compounds. One, to get the patient sleep. The second one, feel not pain to the patients, have the patients not feeling pain. The third one is to muscle to be relaxed in order to make easy the life of the surgeon. Another one is for the mind to enable the patients forget about the surgery. Because it has been discovered tens of years ago that the patients, even though sleeping, after years they had nightmare recalling about the surgery. So now they use all these components in order to get the patients in the best status. But as I mentioned you, there is patient's metabolism on board, so every patient keep the pills in a different manner. And that's the reason we had several sensors coupled with a front end for reading the exact amount of that molecule in the patient's blood, and then giving to typically mathematical model on the pharmacokinetics of the different components before deciding how to adjust the pump for the next infusion. This was the rationale of the project. Here you see the successful detection of the propofol, paracetamol, and midazolam. There are three of the main components of the anesthetic cocktail. Just to mention, the propofol was the killer of Michael Jackson. I just mention usually not only to make recall to Michael Jackson, great artist, but also to mention how an anesthetic cocktail is a dangerous set of molecules. So they are really bringing people close to that. That's another reason why we would love to monitor exactly patients by patient. And this is another lovely data I have when you see semi-continuous detection of simultaneous detection of paracetamol, also called APAP, depending on the side of the ocean you are, and the propofol. So you see, we succeeded in measuring simultaneously the two components and with no interference each other by using the same system. This is the latest news we have published a few years ago in the anesthesiology. We have integrated everything in a very handleable device. You see here, we have kind of needles sensors that are thought to be directly injected in the patient's vein in order to recalibrate time to time the injection system. So in principle, the anesthesiologist may have one of these devices in his pocket, go to the patients during the surgery and make a one-shot measurement, for example, of the propofol, and recalibrate the machine for the further injection. Just a little note, the propofol is typically prone to stick to the electrodes, so we have the problem of so-called fouling, because in the electrochemical detection, we split the propofol in components, and one of these components got stuck to the sensor and still keep going, giving signal, which became horrible for automatic recognition of the concentration of the propofol. So that little boy also contained a machine learning algorithm that read out us from the problem of the propofol. If you're interested, just let me know. We had a publication on biosensor and bioelectronics. Wearable, I'm sure all of you are interested in developing wearable or in contributing to considering wearable for health measurements. Once we are out of the skin, eventually with the ontophoretic, we can integrate electrodes in our patches and then easily electronics. This was just one of the first prototype. You see here, ceramic, commercially viable electrode that then got functionalized for glucose and lactate for sport monitoring, and we have done here flexible electronics units in order to be carried in a typically pocket for a mobile phone. This is another very interesting experiment we have done years ago, measurements of nicotine on the skin. This was an industrial project. The company was interested to evaluate if it's possible to measure nicotine on the skin of people with the aim to adjust technology for enable people stopping smoking. They had no idea if it would possible. They asked me to have a non-invasive method because not to blood sampling. And so we start asking ourselves if it would be possible to measure the nicotine on the human sweat. We first discovered that the nicotine is electrochemically active compound, so easy to measure. And then we have succeeded with a couple of folks in the lab to distinguish EV smoker by light smoker. And the light smoker here was a former PhD visiting in the Ali and Mehmet lab that time was postdoc in my lab. So I was so thrilled to distinguish EV and EV smoker fairly well with the calibration, because it was the proof for the company that we can just, produce patches to be placed in the shoulder of a person that wants to stop, and then eventually command a drug release, typically nicotine release patch, in order to provide the nicotine to the patients by setting down in the mouth in order to let the patients to read out by, typically will or smoke. This is actually the guy that developed the pen for the propofol I mentioned you before, that was used as a material for another project. We have measured sodium and potassium as a marker of physical effort. And within the frame of the same project, we have also measured the lithium, that is very important to monitor bipolar disorder. The typical problem with bipolar patients is that, they come back to the doctor with the same symptom after month, year of treatment, and the doctor does not know if it's because the treatment doesn't work or if because the patient is cheating. And I'm sorry to say, I had the occasion to live closer to one of these patients. They are prone to cheat because I don't know if you know, but the bipolar disorder is bringing them from depression to high excitation. But in the high excitation state, they feel like being, king of the world. They do everything they want to be the clever scientist in the world, the clever artist in the world, the clever man or women in the world. So when they take the pill typically set down the wave. So they don't feel any more depressed, but they even don't feel any more super excited. And I'm sure you understand me, all of us, bright scientists, are usually super excited of our work, our results. So now we can understand a bipolar patients that feel normal, not good at all. So they stop using the therapeutics And clearly, when the symptoms come back again and the doctor asks them, say, yes, I took the pill. So the problem is that the specialists in the field are highly asking all of us, including you, to develop smarter technologies. And this was one of the smarter ideas we had. So just a patch that you can place whatever you want, and then measure the content of lithium released on the skin by bipolar patients. It was a very well received results by the experts. So if we want to move from wearable to implantable, the challenge is what you put under the skin. Because if you put this under the skin, will be not enough comfortable, right? So we need to minimize what we put under the skin, and then we have more freedom with what we put on top of the skin. You know, we can put, I showed you right before, we can have in a pocket for a mobile phone enough electronics to be then connected with mobile phone, whatever, Wi-Fi or Bluetooth. But the real challenge is what we put underneath. So no battery if we want to scale down, and then eventually transmit power and receive data. So this is one example of the wearable devices we have done for implantable. You see here fairly large antenna to transmit power and receive data, and the typically commercially viable Bluetooth communication module. And then, as I mentioned, the challenge was in the implant. So receiving the energy by an inductive coil, so typically by magnetic induction, then realizing integrated and ASICs, so dedicated electronics, for our sensor that in this baby was on the top. So we have then sandwiches all the layer and then provided wire bonding, and the aim was to stay close as possible to 1 centimeter square and very few millimeter thick. So here you see the baby, and you see also the biocompatible packaging we have put it. And this is exactly the toy that we have inserted in the mouse you have seen in one of the previous slides. So with this device, we have monitored continuously glucose and paracetamol as a proof of endogenous biomarker and exogenous drugs in precision medicine. And eventually the question is that if ever humans will accept this under the chip, even though they are small and minimally invasive. So I always queried my classes in teaching. They always send me clear messages, not really. But a recent survey done in Europe, you see 2022, so a couple of years ago, turns out that half of the population, whatever age, is ready to accept under the skin devices, not only for health monitoring, but just for payment. Instead of showing your mobile phone for Twint and the supermarket or your credit card, can just show your arms and have a passive RFID implanted below there. And it seems people are eagerly going to accept that. Of course, if we want to be at the same scale of the RFID that we typically implant nowadays in our pet, they are really big like that, we need to integrate everything in a smarter way. So all our molecular sensor, typical pH sensor and temperature meter, smart, coils underneath, and dedicated electronics. Well, we have done kind of 10 years ago in a scale of few millimeter diameter, so really in the scale of huge surgery needle. But and here you see the device with the biocompatible packaging. So we have done, of course, at research level, but most importantly, Mark Phelps already in 2015 showed us the new ECG monitor devices that Medtronic has introduced in the market a few years later, the new Reveal Link, which is at the moment only an ECG monitor, but you see is inserted like the RFID of our dog, just with a syringe and with 1 dot of suture. So not any more surgery to get under the skin electronics, but just injectable by a syringe. And Mark told me they are, of course, thinking to bring on board sooner or later also glucose monitoring. So what is the message here is that if big worldwide leader in implantable electronics work on syringe injectable electronics, it's clearly a future that is ready to come. Once we are there, as I mentioned, we go to Wi-Fi. We can show, lactate, glucose, paracetamol, whatever molecule we are capable to ready, we can then retransmit. And most important, we can reach the connected swatch of whatever intensive care unit responsible or whatever boss of any hospital in order to make alarm. You see name of the patients, what is the molecule out of range, and what is the range of the molecule. So it's done. So we can resign the contract of all of our academics, and because everything is done. So now the question is that what else? Okay, industry now is working to bring to the market, but what we're doing in the Terasaki Institute of Technology, for example, to look at 10, 20 years ahead from now. Because if wearable is now in the age of industrial transfer, unless we are a research-oriented, an industry-oriented research center, we need to look to something that now maybe look like science fiction and in 10, 20 years would be maybe reality. So what else? Okay, I accepted the challenge several years ago, and I thought about to investigate the concept of the body dust, which is in some extent, if you want, an extension of the concept of the smart dust that has been already extended in the concept of the neural dust. In both the cases, University of California, Berkeley. So what is the concept of the body dust? That's an idea that came actually several years ago with a friend I have again in Imperial. The idea was to think about an electronics that could be spread automatically in the body just by drinking. Not as swallow pills, that's already in the market, but something that go to the bloodstream, which is the challenge. On one side, for example, Gartner forecasting for hypercurving technology set smart dust as a next incoming technology. Actually, IoT, in my opinion, is a correct translation of smart dust that is already in the market. But my point is what to do for the body. So what do for the body, as I already mentioned, is let us imagine as a vision to have a person, not necessarily a patient, or might be a potential patient, that is just drinking a glass of water. And then in this glass of water, there are so negligible size electronics, diagnostic electronics, that could not only go, you see in the cartoon, in the digestion tract, but pass from the digestion tract by through a phenomenon that is called internalization. So our intestinal tract has the capability to take a cell, internalize in the Caco2 cells wall, and then to spread in the bloodstream. So once we are in the bloodstream, of course, we can imagine to, for example, freely flow all around the body through the bloodstream, and eventually meet, for example, in an oncological patient, a tumor mass, and let these particles stick there if they have been functionalized with, for example, antibody that track the surface antigen in the cancer cell, and then provide, of course, telemetry. So if we want to envisage that, if we want to joke a little bit with science fiction and try to address seriously, scientifically speaking, that challenge means we need at least to go with the size of the blood cells, in between 7 and 30 micron. So let us see if this is a mission impossible or if there is something we can do around there. So we need extremely small sensor. We need extremely small electronics. First for the sensor front end, but then we need also smart and small electronic for data transmission, typically for data link. We need to find a smart way to power all of this stuff in that tiny sizes, and then we need to demonstrate. These are the 5 main steps along the way. So let us see what is state of the art now. Hirleman Group, ETH, have a look of the date. It's old stuff, 10 years ago. They succeed in integrating. This is again nano platinum, directly grow on top of working electrode of a CMOS, exactly what I showed you 15 minutes ago. But the group of Hirleman succeed in measuring glucose, reliable glucose measurements down to 5 micron working electrode, below 10 micron. Okay, it seems is feasible. Then, of course, we need electronics that scale. Electronic for reading the biosensor, for the glucose, for the drug, for the lactate, for the potassium, whatever marker we want to measure. Well, already in 27, we have actually published a design where we succeed in getting a potential stat, so all the electronics we need to read out the current from an amperometric sensor in 10 by 10. Honestly, it was only the design, and it was only the design of only the potential stat. We need other service electronics. More recently, this was done in collaboration with Imperial College. You see, again, my friend Pantelis Georgiou. More recently, with Paolo Crovetti and other giants in analog and digital electronics in Politecnico di Torino, we have instead designed a completely different potential stat based on digital electronics. He found a way, kind of 10 years ago, to redesign in time domain circuits that perform similar to operational amplifiers, but they are, as you see here, just done by shift register and logic gate. So saving a lot of area. And with that, we have layout 20 by 20. We are not still at 10, but we are very close to 10. 20 by 20, and the baby has been sent to the foundry, already received, is now under test. What's about the data transmission? Well, we have done years ago an unfortunate case. You see, it's just published in an archive. We didn't succeed in getting published. And the problem is that, it's still big. And the reason why it's big, because the starved ring require capacitor, sorry for the electronic details, and usually capacitor in CMOS design is more hungry in terms of areas. So years later, we set down the technology. You see, the previous technology was 0.18 micron, quite old. We moved it to 28 nanometers, and we succeeded in avoiding to have explicit capacitors by exploiting parasitic capacitances. And the guy was 5 by 10 by design. Then, thanks to this idea, we went back to 0.18, and we have recently taped out, received, and successfully tested. It will be presented next June in MOCA's conference. You see the scale, 10 micron. This is our oscillator. This is our oscillator, which is the main hanger in terms of area components of whatever data link. So it seems we are getting closer again in 10 micron with technology still fairly large, 0.18. So we have fairly plenty of room to set down with more modern CMOS nodes in setting down the sizes of the electronics. What's about power? Well, this is the work I mentioned from Berkeley. They did it, and they already provide tests in mice for neural dust. This is the stimulation of the sciatic nervus, but they did in 1.7 millimeter cube by ultrasound powering. It's several orders of magnitude too big with respect to our needs. So we went back to literature, and we discovered that another good friend of us, Ralph Etienne Cummings, already did years ago with 200 by 200 micron coil receive enough power for a stimulating brain electronics. So we started from that work, and we imagined 3 scenarios for our body does. So translational medicine, where the mouse is small, so the electronics will be close to the surface of the transmitting station, or in vitro diagnostic, we can mix with our sample and then put the plate on a powering device, or, for example, heart monitoring, where the organ is so close to the skin that you are not very deep in the body. Well, in those cases, you see decent amount of power in few micron few micron watts power scale seems to be not that big, 150 down to 65 micron. With the digital potential stat by Paolo, we are at 9 nanowatts. So we can really again go down 2 orders of magnitude or 3 with respect to this coil size. So, because this was driving us around 150, we started from under 50. This is one of the latest baby we have received. You see here the coil is realized not anymore with metallic thin film, but by exploiting the metal layer inside the CMOS coil. So it's received directly by the foundry, and inside, you see here inside the area of the coil, we have all the electronic we need. We are trying two applications. One is free floating for glucose monitoring or dopamine monitoring in the brain. The second one is for neurostimulation. Actually, this version you see inside the electronics for the neural stimulation. But it seems feasible. We have already tested the received power by the coil, and we receive enough power close to a few micron watts to power all the electronics we need in 150 by 150. We have also developed smarter idea to receive more power by moving from a passive resonant coil in between the transmitting and the receiving. And you see here an electronic IC that shifts the frequency in order to be more reliable with more power receiving in the first layer when we pass through the skull, and then go high with the frequency in order to reach the different implant. And here is the implant we have realized in EPFL. You see the scale is, 150, as I mentioned you, of lateral size with two very long stimulating electrodes that went deeper in the brain tissue. This baby is thought to completely restore vision to totally blind person. The idea was to have a camera in the glasses, a transmitting system here, and then thousands of these dots spread in the visual cortex, which is in part exposed in the back side of the brain. So, along the challenge as set it in 21, it seems we are doing quite well with the sensor and related electronics. While we are approaching also small size with the powering, I have to confess you that I'm also exploring more recently other two options, biofuel cell, in order to retrieve energy directly by the glucose in the human body. And the second one is micro batteries. We discovered a bright scientist in Dresden in Germany that is working with micro batteries that has a different principle that fuel cell, but seems to be capable to power with really small sizes. And then, of course, we are still away for a in vivo demonstration, but I'm quite confident it will come. So I'm at the end of my talk. I just moved from this joke from the newsletter Time that, a few years ago, 2019, if I'm not wrong, they were challenging us and say never offline. So having electronics, monitoring electronics on our skin with, a screen in our skin. Well, that's, again, apparently a science fiction vision, but as we have seen with body dust, there are serious people all around the world working on that. And so we can easily imagine that we still need to design dedicated electronics. Yes, okay, we still need biotech, a company providing us smarter, typically biological proteins that can survive for months or years in our sensors. We still need nanotech company providing us smart material in order to improve the performance of our devices. But if we bring all together, it seems very simple, close to easy to realize portable, wearable, and implantable device, and maybe one day also spreadable diagnostics in our human body. Well, as you can imagine, you have an enormous depth of gratitude to all the people working with me in EPFL. This is just one of the last shot with the people at the moment in the lab, and I thanks all of you for your kind attention to my talk, and I'm happy to answer any question in case. Thank you.
Thank you very much for the great presentation. I have a couple of questions for these dust sensors.
The brain?
Yeah.
Okay.
What is the substrate?
The substrate of what?
That's CMOS.
So the substrate of the CMOS is a silicon wafer.
Yeah.
That usually they are several hundred of micron thick. So we use typically chemical etching to reduce to 30 micron. And then I have to be honest, I haven't had time, I apologize to show you, but if you want to have details, how to get these tiny electrodes, we typically build on wafer scale, So they are built like, L's with a chip in between. And then we remove everything, and then we bend the top and the two stimulating electrode to penetrate the brain cortex.
And for the fabrication, you use focus ion beam?
Well, yes, for the lithography, because we are really down. So we use normal lithography, but we create the mask with the focus ion beam, yes.
So the question is, how feasible is that? Because for each.
Good question. So the plan is to create thousands of these pixels. You have to imagine one of this boy like one pixel in an image. It's much way more complex than that, because the visual cortex does not one-to-one match the image like in a computer. But let us keep the details for a while. Let us imagine I want to stimulate that bundle of neurons. Then the idea is to pick and place. So I had several discussions with experts in vision that I use it also sometimes to do it also for the retina when they build the system. So in principle, it's feasible to have a robot. Then you open the scalp of the patients, and then the robot cut and paste. It takes half an hour, 1 hour, but at the end you will have 1, 2, 3,000 of these dots inserted gently in the cortex. We have also done at the moment just simulations in order to create the electrode tough enough to penetrate, because our first prototype, as you can imagine, was destroying when we tried. So now we have done simulation and emulation in agarose, so using agarose as an emulation of the cortex, and we have successfully demonstrated that we can insert. So pick and place seems feasible.
But what happened after? So the silicon oxide and like oxidation doesn't destroy the chip?
Well, no idea.
Okay.
So I love this sentence I have received from the movie Apollo 13. This is the problem 3557. We're still asking ourselves how to solve the number 3. So yes, we received the question, you're right. So my typical answer here is that first, biocompatibility is not an issue. Biocompatible membranes solve the problem. Yes, in brain stimulation, we know very well tissues grow on the systems and then performance went down. That's the reason Tim Davison already 10 years ago spoke about windows open in the brain, because the idea is to retune the system in order to become more powerful. This is in place with the present technology, and the market will be not scared by this approach.
And the last question, do you think, like technical wise, can we do that on the conductive polymers instead of silicon substrate?
So are you suggesting to use conductive polymer instead of CMOS technology?
Instead of just silicon, because we can print on that too, right? Can do the lithography. Well,
in principle, yes. In practice, depends. What is in my opinion, what is the real application and how deep you want to play? So, for example, just to understand each other. If you want to have on board the same number of functions you usually have with CMOS technology, my answer is forget it. Literally forget it. So it's possible in principle to realize polymeric transistor. You also have done a bit of research on that or collaboration, but CMOS technology is so powerful nowadays that if you need whatever function you can have in 1 millimeter square or in 100 micrometer square, no chance to get it in polymeric. If you want something simple, something,
biocompatible, this issue is I'm not sure how we can just have like the single crystal silicons as a non-toxic material inside the body.
Yes, I agree with you. Polymer can address biocompatibility, but the problem is the complexity of the function. Just to let you understand, our single dot need to receive power and data. And data contain a word, and that word tells to this dot if he has to fire or not. A lot of digital function need to demodulate the signal, recognize the byte, go inside the byte, recognize the bit, then have a logic that tell, okay, shut up or fire. So you see, that's really a lot of complexity that in CMOS you can have, I'm not kidding, for free. So because the CMOS cost for 100 micron dot is negligible nowadays with all technologies. So, and it's an enormously robust system. So biocompatibility, that's an issue, but at the moment we have biocompatible polymeric packaging that protect tissues by the implant, so why not using silicon or whatever other metal that assure us robustness and then package in a way that is biocompatible. It's a different discussion if we want to just do surface electrode. This is a completely different discussion. You can do probably enough reliably surface electrode for the cortex with polymeric material. In that case, I'm totally in favor.
Your sensors are electrochemical 3D. Have you looked into FET sensors?
Well, that's a good question. Yes, of course, we can do it. I'm not that much expert in field effect transistor sensor, but for example, for ion sensing, they work very well. For sequencing, you know, uTorrent, now Thermo Fisher exploit a uTorrent invention, which is actually an Imperial College invention on sequencing based on FET. So this is a fantastic technology. It's state of the art in CMOS, so you can do billion of sensors in a wafer scale. So cheapest, reliable, of course, not good for all the detections kind, but for many, for example, ions is very good as well.
Thank you very much for your great presentation.
Thank you.
I want to know about the material of chemical structure and composition of your wearable sensor and what's its basis on. It's on PT or the other material?
Well, honestly, we have done it with many different kind of approaches. So we have used polymeric substances, and then we have used even polymer electrodes, polymer-based electrode, typically to measure on the skin. I showed you in the opposite case, just ceramic-based electrode, bought and used it. So in my opinion, again, depends by the application. If you are in a region of the body where the conformability is a key, then flexible substances like polymeric substances are great. That's the reason I mentioned for the surface electrode in the brain, I'm totally in favor. They are soft, they are biocompatible, working well enough. And then you can always couple with electronics we have done. So we have realized for wearable sensors, flexible substances on polymeric material with electrode, and then we have coupled with, you know, normal CMOS electronics. If you are instead in region where they are very flat, you don't need anything special, you can even buy, you know, even rigid electronics and rigid electrodes, and then eventually integrate in a soft mesh. So you can have a soft wearable device done by very hard pieces of, you know, hard material that the people even don't feel uncomfortable. So there are nowadays literally many different approaches. I've seen over the last 10 years, organic electronics growing a lot. So that's really up to a choice in terms of performance in one side and costs in the other side. Don't forget that it's very different, in my opinion, the cost issue if we think to an academic or a research prototype versus an industrial one. A company wants to have supplier, and supplier means reliable supplier, not startup that today exists and tomorrow may disappear. So, and of course, cost means cost for billions pieces a month or a week. So that's the reason sometimes I think if you need high level functions, low cost CMOS electronics is better than whatever polymeric that is, you know, not really with high function in the market yet.
So much.
You're welcome.
You're using rats for your samples.
Again,
You're using to test your chips or dust into small animals. I saw a piece of a rat, picture of a rat. Harbi's group, they were using African beetles, which you don't even need animal protocol. You can just use it. Have you thought about using insects or other things to test your neural dust?
In vivo, you mentioned, right? Sorry. So far, we have done in vitro. We plan to do it in vivo in collaboration with Bern University. They play with monkey. So the brain is enough large, and they usually use with respect to our babies, they use enormous electrodes. So I'm not on that side of the research, so I cannot disclose details because I have no idea about the details. What I can tell you is that we have done some preliminary work with them, with larger electrode, just to set the right function in our CMOS dot for the right simulation. And this seems to be, easy done in the lab, even with the monkey fully awake, because they typically wants to register reaction by the monkey during the simulation. So it's fairly scaring, but they keep the monkey, they block the monkey, they open the scalp, they put the electrode, and they let the monkey, for example, react to what she sees on the screen and then collect the data. So we plan 3 to 5 years from now to set similar experiments.
Hello. Thank you for a great presentation. I have
Thank
a
you.
question regarding your wearable sensor for monitoring the level of lithium in bipolar patients. Question is, if by any chance you figure out the correlation between the level of lithium and the serotonin or cortisol level in the patient?
The answer is no, because we didn't thought about that, because we have been not required about that. But there are technology for doing that. So, for example, dopamine is fairly well established in literature. You can just use, microneedle electrode to measure the dopamine. Pedram actually did it 15 years ago in mice. So you can take a rat and you put, few electrodes in the brain, and then you can monitor the dopamine. I'm not sure about that, but might be we will start a new project in collaboration with the University of Lausanne, where the plan is to measure glutamate and GABA. GABA is a metabolite from the glutamate. And there are now data, not in bipolarism, but in autism, that say if there is an imbalance between GABA versus glutamate, the baby is going to develop the autism. So we can use similar technologies. If you think our dot with the 2 antennas, we are already designing electrochemical sensor along the antenna. I didn't mention, but the antenna is very long, so several hundred of microns. But the stimulating region is only on the tip. So we have a lot of area, lateral area, where we are now depositing electrochemical sensors. And the idea one day would be to record electrical or stimulate electrically, meanwhile measuring typically neurotransmitter. So including dopamine or whatever other molecule in the brain that might be correlated to the disease, bipolar, autism, or whatever other mind disease. So not yet, but work in progress.
Thank you. And the other question is regarding the screening of the patients. And which patients actually included as bipolar patients versus those are suffering from the depression?
I'm surrendering. No idea. I'm not a clinician, so I'm on the side of developing the technology.
Or it was based on the...
No, I really have no idea. So they just asked us, the wearable technology to measure the lithium, and then, we did it. So we are not yet at the stage, if you like, to use the technology in a real trial. And once we will be there, will be not me selecting whatever stratification. So, for example, I can offer you another example in cancer research. We had designed nanowires by exploiting a funny phenomena, the memristive phenomena, to detect several cancer markers. And then we give the technology to a biologist, a biochemist actually, in the Ludwig Research Institute in Lausanne. And she stratified the patients with the normal technologies, and then she verified that the stratification done with our technology was bringing similar results at lower cost and more faster results. But that usually that's not me, that I take part on how stratified patients or selected patients. So I'm sorry, I cannot answer. In case I can bring you in contact with experts in this kind of diseases with whom I'm collaborating.
Thank you so much. Appreciate
You're
it.
welcome.
Thank you, Dr. Carrara.
Thank you.
I had a couple of just questions dating back to your first set of slides. You shared some impressive sensitivity numbers on some of these sensors. What about specificity? Is that a factor, and are you challenged in any way on the specificity side?
Yeah, thank you for the question. Is the question, is one of the three questions, major question in biosensing. So, very good question. So usually the selectivity or specificity, so it's more correct to call it selectivity, but all of us, including me, call it specificity, because in the case of the glucose monitoring, is correct to call it specificity. So the selectivity is usually done by the biological probe. And here, the two sides of the coin is biological probe and the selectivity of the surface. So let us start from the first one, selectivity of the probe. So in case of oxidases, this is 100%. That's the reason we call it specificity. So if I use glucose oxidase, I just get data if glucose is there, unless I'm badly designing the system. In case of the cytochrome, we are close to health, because I don't know if you noticed, but I showed you two different detections with the same isoform, verapamil and cyclophosphamide. What does it mean? It means that if I have in my patients the cyclophosphamide and the verapamil, my sensor will detect both, because the enzyme has both as the substrate, and some enzymes has many different substances. So in that case, one way we are using now in order to increase the selectivity are, if you like, a twofold strategy. In one side, we try to design the surface in order to distinguish the Faraday peak, not to have them all overlapped in the same potential. And I have, lovely data by using many different kinds of drugs. If you're interested, I can show you. And then, because the interaction of the different substances on the same enzyme are not trivial, more recently, and I got the news this morning, we got accepted in Electrochemical Acta with one of first paper on that. We're using machine learning to distinguish the different components from the different drugs. So we, as I mentioned before, we do not rely on chronoperimetry, mission impossible. We need to scan the voltage, acquire a kind of voltage spectroscopy, then give this signal to a machine learning algorithm, which has been trained on thousands of sets of drug detection, who recognize where is the peak and what is the drug involved in that peak. Something similar, but very different than what we have done years ago with the propofol, where we have used the machine learning machine to follow the peak shift due to the fouling. Now we follow the shift due to the interaction with the other drugs. So complex mission, but seems affordable. So,
thank you.
You're welcome.
And maybe related to that, have you done any sensing of hormonal levels? Hormones?
Oh, good question as well. No, but this is by chance. So I'm considered myself, like a shopman. So if you enter my shop and you ask, I'm more than happy to study with you the technology or the sensor. And usually, my shop is entered by doctors or biologists or biochemists. So I had thought several times in free discussion with friends and colleagues about hormones. Several isoforms of the cytochrome P450 detect hormones, I didn't have time to mention, but there are many other detection techniques based on aptamers, on antibodies. So if there is no an oxidase or an enzyme or a cytochrome, we can always look for other enzymes or for other biological protein. But in my direct experience, I have not done yet, maybe in future, because I know
Right.
they are interested in molecules as well.
And one more question. I'm just curious on your nano or your dust type of particle. How do you ensure that something that small, once it's inserted, stays where you want it to be? And also, what's realistically the time that you envision something like that to stay in before you start getting, inflammatory responses or other things?
Yeah.
And then how do you remove it?
Yeah, that's another good question that brings us back to the biocompatibility issue that was asked already. That's a very good question. So there are several ways to answer. So one is that if we electrodeposit, as for example, the nanoplatinum that me or Hilleman or others are doing now, this is, chemically linked. So it's like, our mental artificial teeth, yes, but they are really thought to stay forever. You need to, to beat on gold to get pieces of gold away. They are called not by chance metal, noble metal. If you do with carbon nanotube, that's a completely different story. Carbon nanotube or other kind of flying particles, they might be dispersed. Again, as I answer her, we are back to the biocompatible membranes. I remember to have done years ago in collaboration with IMEC in Netherlands. In the Netherlands, there was a group expert in biocompatibility of implantable electronics, and we had the suspect to disperse our carbon nanotube. So what we have done was to package all our system, including carbon nanotube on top, that they were not chemically grouped, but just dispersed, so weakly linked, but then block it with typically polymeric matrices. We have done experiments in vitro with cells, typical toxicity experiment, and we passed it very well for 3 weeks, 1 month of application. There were no dispersion. Then the experiment you have done with the mice, the chip was implanted for 1 week. The chip was explanted, the poor mice was killed, and also measurements of typical inflammatory reaction has been done. We have tried 3 different packagings, and in all the cases, the inflammation was under control. We saw only the inflammation coming from the initial surgery, then going down after a couple of days. So it seems we can do it, block it and not disperse, at least for enough short terms. 1 month of experiment done so far.
Thank you.
Thank you.
Yeah, fantastic talk. So just a kind of question, I'm a biologist, so I'm really kind of naive on the biosensor kind of front. So kind of my first question regarding to the dust sensor. So if you really kind of in the future, people can drink it. So how can you really kind of control, how you guide those sensor to the particular organ or particular locations you want? So, yeah.
Good question. So actually, the powder is thought to be not controlled for that.
Okay.
So nowadays, if I have an inflammation under the skin here, I take a pill there, and the human bloodstream allow me to bring the drug there. I have a lovely example because it happened to me as a patient. Inflammation of the shoulder. I'm sorry to say, but approaching 60 happen more often. My doctor told me, forget it to take anti-inflammatory pills because the vascularization of this joint is so poor that your pill will never reach it there, right? So it's not a problem of the body dust, it's a problem of whatever drug delivery system. So what I'm targeting is two aspects of the problem. The first one is surface covered. We are not yet there, but there is simple solution. Surface functionalization, in order to be sure that if I want to target a certain kind of cell, my dust will stick there, of course, once it will meet the disease. And the second is how to secrete all the particles that didn't stick it there. And this brings me to the billiard system, back to the billiard system. And if you like, the bad news is that there are experiments that show that bacteria can move from the bloodstream to kidney and to urine, but they have at least tested at 7 micron. So not 10, but 3 micron less. So yes, this is my challenge, if you like, to do so tiny diagnostic dust that could be, in the worst case, just spread out by the billiard system.
Okay, got it. So kind of another question. So regarding the lifespan of those biosensors, if it's enzyme metabolic based, so I don't know, just like how long? And, yeah, and how many times those kind of the subject and the enzyme, right? So kind of.
Yeah, that's another fantastic question. Thank you for that. That's fantastic because there are several bad news on that pathway. So the first bad news is, if I plan to use the cytochrome, we have published very clearly years ago, semi-continuous detection of the naproxen in vitro, 15 hours, 1-5, and the enzyme is gone. So we have really registered very stable data measurements, and then after 15 hours, we typically see the exponential decay of the performance of the sensor. Not very good for long-term application, right? But if we look at the case of the glucose oxidase, we did in our lab, even with wild-type versions, up to 44 days. But 2015, if I'm not wrong, 2015, there was a publication in pigs, continuous monitoring of pig up to 1 year. What is the difference? Engineered protein and engineered ways to keep the protein on the sensors surviving long, like in the human body. In human body, all these proteins are key for the metabolism. They are thought by the nature in order to be changed frequently. So the P450 is changed on daily basis. Every day we have a new set of cytochrome P450, like antibodies. But biotech, not by chance, I've mentioned in one of my last slides, we need help from the biotech, can of course develop engineered protein much way more robust than the native one. And in that case, of course, we can extend the lifetime of the sensor, because clearly the biomaterial is the weaker part that affects the lifetime.
Thank you.
You're welcome.
Let's thank the speaker one last time.
Thank you all.
Thank you for your time.